Great. Okay, so I'm going to change tack, and I'm going to talk about not evolution of species, as we've heard in the last couple of talks, but I'm going to talk a bit about a different sort of evolution, namely the evolution of the T cell repertoire, which is a rather special case where no mutations really occur, and we're really looking in isolation at selection on a big population. So I'm aware that the audience is pretty diverse. So just a little bit of introduction to the T cell receptors. So T cells are the white blood cells that are responsible for a lot of our immunity. Let me just change this pointer so it's more visible. There. So here is a structure of a T cell receptor at the top of this picture here in green. So imagine this is on the surface of a T cell, which is up at the very top of the picture, and it sees antigens derived, let's say, from viruses. You know, this could be a little piece of COVID-19 or flu or something like that, which is held by a receptor, the MHC molecule. And you can see it interacts with it. And when this interaction is of a sufficient tightness or strength, then you get a signal to the T cell, and it can do one of many things, but it basically tries to deal with this antigen that it sees in some sort of appropriate way for the body. I'm not going to talk about that today. And the interaction is mediated by loops, protein loops. There's 6 loops altogether called the CDRs, complementary determining regions, and there's 2 chains and 3 on each, so there's 6 altogether, and they come down to make essentially a surface which interacts with this peptide binding structure there. So the way that the immune system can recognize so many different things, essentially anything that you inject into the body in the form of a protein, or in fact many other macromolecules as well, but I'm going to focus on proteins today, can be recognized. And the reason for that is that there's a system for making millions and millions, probably in the 1,000 million, slightly different receptors with a slightly different sequence. And these are made, obviously, we have in the order of, I think, around 10 to the 9 or so T cells altogether in the body. I mean, 10 to the 11 also T cells in the body. And since there are about 10 to the 9 possible T cell receptors, each one is probably present, you know, there's about 100 T cells with a particular receptor at the start, but it's not a uniform population. Some are made more and some are made less for various reasons. So if we think about the evolution of the T cell repertoire, so here's just a representation, don't take this picture too literally, but imagine that each of these dots is a T cell receptor, and the size of the dots is a measure of how many T cells in the body are carrying this particular receptor. And as I say, it's a non-uniform distribution. Some will be a lot and some will be a little. And here it's just reproduced in a 2-dimensional space. And you can see some areas will be quite full of T cell receptors, and some areas will be relatively empty. And the idea of adaptive immunity, in a way, is that if a pathogen arrives, it has to kind of penetrate. If you think of this as a kind of a mesh rather than lots of dots, then you can imagine that for a virus to infect a person, it has to sort of get through this mesh. So if it chooses a place where there's a lot of holes, it'll stands a good chance of doing it. But if it bumps into one of these receptors, then it won't make it. So imagine this area here, and then you're exposed to a particular pathogen. You get infected by flu, let's say, and then what happens is that area of the map becomes much, much more full, because these T cells, which were here, which recognize that particular pathogen, divide. So you end up with many more T cells carrying the same receptor. And the same is true actually for B cells and antibody, but I'm just focusing on the T cells for the moment. And so, so what we're interested in knowing is, can we define, so I've shown these in some sort of arbitrary space, but can we really define two T cell receptors that are similar in this space, in the sense that they will recognize the same pathogenic challenge, simply by either their structure or, even better, by their sequence, because it's relatively easy to obtain DNA sequence for these T cell receptors. It's possible but harder work to obtain a structure. But even given a structure, can we really say by looking at a structure what constitutes a similarity metric between any two T cells or a set of T cells receptors? So these are the questions that you might want to ask. Could we recognize a bunch of T cell receptors which recognize the same antigen within or between an individual? This might be useful if you're studying the immune response. You'd want to know, you see a set of TCRs and you see them again in somebody else. Would they have some sort of similarity? And also a similar question would be, if you change the antigen a little bit, could we predict which T cell receptors are likely to recognize that changed antigen? And as I said before, we can do that from, maybe we can try and do that from if we had the actual structure of the molecule, but it would be even better if we could do it just from the primary sequence, or just from, because that's an easier piece of data to obtain. And as a starting point, classical measures of protein distance don't do very well in terms of alignment. And you can probably imagine that because just of the loop structure that I showed you before. But even if you focus down on those loops, these measures, simple measures, don't work very, very well. And it's not immediately obvious what constitutes a good metric. And so why might this be? So I'm going to try and illustrate it here and show you a little bit of data about this. So here's a picture of a T cell receptor bound to an MHC peptide. You can see the peptide is almost entirely hidden because this is a good, an efficient binding. And also the peptide is held quite deeply in its MHC peptide binding structure, so you can't really see it. Now, and here's the two chains of the T cell receptor and the loops coming down to touch the surface. Now, if you pull this structure apart like that, so I've just literally pulled the structure apart, and you can see here in color, here's the peptide, here's the two chains of the T cell receptor, and here's the MHC. In pink are the residues on the T cell receptor and the peptide, which are in close contact, I think below 5 angstroms or something like that. And you can see the critical point that I'm trying to show with this figure is that the interaction is very much a hotspot of interaction. You get a contact hotspot. It's not an extended interaction over a large distance. So that's why when you want to think about metrics of difference, you want to be looking at something that can look at these hotspots of distance rather than a kind of global measure of a relationship. So I just want to show you a very little bit of data which has been prepared by Martina. So there are quite a lot of actual structures now in the PDB database, about 180 T cell peptide MHC structures. And she has fortunately didn't have to actually trawl through the PDB, but somebody's done that already, and there's a special database just for T cell receptors. And what Martina's done is to try and extract the interaction information between the TCR and the peptide MHC. And this is how we've tried to do it. So what you see, let me just explain this figure a little bit, this heat map. On the left, you see the peptide that is bound in the MHC. So this is the antigen going from amino acid 1 to 13. In general, these are in the order of between 9 and a little bit more than 13 in length, so short peptides. And then along the bottom is the standardized numbering for the T cell receptor itself, and particularly the loops, the CDR1, 2, and 3 loop. You notice there are white spaces. That's because different T cell receptors have different length loops, and in order to keep the homology together, the standardized numbering makes sure that the conserved portions of the molecule get the same number. So the ends of the loop get fixed numbers, but the central ones, in this particular case, these amino acids simply weren't present there. And then, so here, what you see shown in each one of these squares is the distance between the peptide residue and the TCR residue. And in fact, it's the closest distance of any atom within the residue. Of course, each residue, each amino acid is made up of many atoms. This is the shortest distance between an atom on the peptide and an atom on the TCR at that position. And you can see there are, again, hot spots here, just exactly as I showed you. This is the alpha chain and the beta chain. And we've represented it a bit easier, I think, to see here. These lines are everything that is we would consider a contact. Again, I think the interaction threshold is 5 angstroms in this picture. And what you can see here is this idea of sort of hotspots and sort of short motifs interacting. So, for example, each line here is a contact, and the depth of color is the number of atoms which contact between that residue and the other residue. So this position 110 here, for example, you can see it has interactions with these 3 or even 4 amino acids on the peptide. All make at least one interaction between the TCR and the peptide. And so there's a hot spot of interaction here on the alpha chain around here, and another one at the end. And here on the beta chain, again, there's an interaction. Interesting here, for example, you see there's a gap. So there's an interaction with 112 and 110 and not with 111 and so on. So that and you can look at all these in a group, and you can see, I'll just show you 2 examples and then a summary, that there's constancy, but there's also variation. So for example, in this particular TCR peptide interaction, there's almost no interaction with the alpha chain. You can see that up here, and you can also see it down here. While in this one, there's quite a strong interaction at the very end of the peptide with the alpha chain, and there's also this interaction, and some amino acids seem to be able to contact both alpha and beta chain at the same time. So you can imagine this is really very close packing of these 2 molecules, or 3 molecules, if you like, peptide and the alpha and the beta chain, in order to give you this thing. And here's a summary of all the structures in the PDB. For the alpha chain and the beta chain. And again, what I want to emphasize essentially is the sort of 2 sides to the coin. Clearly, the distribution of contacts and how they're arranged on the T cell receptor is not random. There's obviously places where you get most interactions, but at the same time, there's no completely hard, fast rule, which is why it's quite difficult to learn this information from a set of sequences. If you think that this is the number of T cell receptors which have a contact at a particular pairing between peptide and TCR. And you can see that none of them go above 70, although there are 180 structures that we've analyzed. That's, in other words, there are many alternative solutions to the problem of bringing a T cell receptor and a peptide together, but it's a limited set out of the total number of possible interactions. So that's sort of additional information about the hotspot nature of TCR peptide interaction. It's a highly plastic molecule model, which allows many possible configurations, and they all give you a sufficient binding energy to allow TCRs to be driven by that peptide. And just, I'm conscious time's moving on. So in the last part of the talk, I just wanted to get an impression of the way we're now going back to try to sort out this question of a TCR metric. So in our past work, based on this idea of hotspots, we focused very much on the sharing of triplets. So we identified, we suggested the possibility that the specificity of the T cell receptor is held by very short structures or sequence motifs, and we focused on triplets, partly because triplets, there's enough of them to make it a very rich feature space, but on the other hand, there are not so many that it's impossible to analyze. We've published several papers where we've sort of counted the number of triplets in different ways and used them to map to T cell receptors. And if you look at T cell receptor as in particular antigenic stimulation, so for example, this is a set of T cells which are amplified in a lung cancer. These are another set of T cells which are amplified following vaccination in blood in human volunteers. And this is a very recent set that we've accumulated looking at COVID-19 receptors. You can see that if you use these triplet sharing as a measure of interaction, then you get some nice structures, and you can do some nice things with them and look at it. But we'd like to go beyond that, because so far we haven't taken into account, for example, where the triplet is found. We just counted triplets, but clearly there's positional information, and it may be that triplets per se are not sufficiently flexible. We should be looking at some more subtle interaction. And so this is work which some preliminary work has been published very recently in a paper that's just out in eLife, and a fuller paper is sort of in preparation. But we've been starting to look at the idea of capturing these sequence motifs and distances between sequences in using a Bayesian probabilistic kind of framework, where you can imagine, so imagine that these P1, P2, P3, P4 are positions along your T cell receptor, and they the amino acid which is found at that position, and the triplet, for example, which is found at this position, in this case T1, will be conditional both on the position along the T cell receptor, but also conditioned on what's the antigen that is being recognized. So you could imagine that you might have two classes of TCRs, one recognizing COVID and another recognizing flu, and you can build up a probabilistic model where these nodes are probabilities of any feature that you want on those sets of TCRs, conditioned on the antigen which is stimulating, or the peptide which is stimulating the response. And these turn out to be really very flexible and powerful tools for looking at T cell receptors, and we're sort of exploring that in more detail now. So, for example, you can show, going back to where I started, that if you have, imagine again this T cell receptor, and if you think of this Bayesian net the probabilities associated with these sort of networks as a kind of a feature, a map, you can show that there are indeed much more similarities between the TCRs which recognize it. One virus, let's say CMV, I've just given that as an example where we've actually done some work, while another set of T cells is perhaps recognizing a different virus, Epstein-Barr virus, and you can classify them according to these Bayesian networks. So I'll finish there. I just want to say that the structural work that I described was done by Tom Peacock, a PhD student who's just finishing now, and Martina Miligeti, who's a current PhD student working with me. The dynamic Bayesian network work is being done by a postdoc, Tahel Ronell, who's a mathematician by background, in the group. And everything would be impossible without the actual T cell receptor sequencing setup, which has been done very efficiently by Imran Udin in the past. He's now gone on to run the single cell sequencing facility at in our local building, and Gayatri Nageswaran, who's doing the T cell receptor signaling sequencing for us now. I think I'll stop there, and I think we've got some time for questions.
All right, thank you, Benny. I, of course, have many questions, but I really feel bad, especially for you dominating. So maybe I'll start. Do you actually see, like, if you didn't do the triplets, but you just took the alpha chain or the beta chain and the peptide, so doublets like that, would you see them then merging together when you see the triplets?
I'm not sure I follow that question. Sorry, what did you mean?
Ultimately, I'm asking what's the, what changes in your cluster structure if you were to just use one chain versus using two chains and the peptide? Do you actually see some like...
Oh, I see what you mean. Yes, I see what you mean. You mean, does including the second chain sort of alter the clustering of the structure? Yeah, that's a very interesting question, and hasn't been very much. It's surprising that it hasn't been looked at more, because there are a number of big data sets where people have looked at clustering of the alphas and the betas, but not sort of simultaneously.
I would imagine that, yes, somewhat, but I would really be curious to what extent, right? I think it would be interesting.
We haven't, I agree. And we haven't fully looked at it. I think we need to look at it a little bit more. In some cases, it seems as though one chain is very dominant in the interaction and the other chain can vary quite a bit.
Yeah, we've been seeing that too in a lot of data, that, and it's either one or the other that seems replaceable.
Yes, yes. Martina has been working on using using the distances as a sort of a feature set, and then using them in a machine learning context to see whether we can classify T cells to see whether a particular T cells binds a particular peptide or not. If you see what I mean. And rather strangely, I don't really fully understand it, but if you leave out the alpha data or the beta data, you can often achieve almost equivalent classifications, which suggests that the information is somehow hidden in both of those, but it's certainly not so obvious.
There is something that we've seen, that classification is a much easier task than actually naming features, right? So if somebody asks you, what are the features linked to recognition? That's a much harder question, or what's the subset linked to recognition than just classification. But I think it would be really interesting to explore all of this.
Yeah.
Anyway,
Yeah.
here I am dominating the conversation, so maybe somebody else questions. Otherwise, I'm happy to continue. I actually do have one other question. So, you know, there's a lot of this work now coming out trying to link TCRs with single cell data.
Yeah,
and
a lot.
So, I mean, this sort of also goes in, you know, this would be like another dimension to what you're doing.
Yeah.
And do you have any intuition of how that could influence or increase your clusters and your ability?
Well, I mean, our problem so in the past has been that at least for our TCR sequencing, we can't link alpha and beta, so we get two sets as most people, right, for bulk TCR, and obviously the single cell will generate paired data. So from that point of view,
no, I was more thinking about single cell, but other features of gene expression in the cell.
Yeah.
Than the receptor itself.
Right. I mean, it's a very powerful way for looking at differentiation pathways within, let's say, a tissue, because you can use the TCR essentially as it's a lineage marker, right? Because it's so unique. So from that, we're doing quite a lot of that. That's a powerful way of looking at T cell differentiation. I don't know if I can add much more. We're literally we're just analyzing our first sort of sets of single cell data as we speak. So I can't give you any actual examples of having done that and learned something exciting from it. But yeah.
Okay. Any other questions? Yes, question. Good. We have to read. From Barbara Bravi. Is the search for shared triplets limited to the CDR3 loop only? Are triplets in the CDR3 sufficient for good clustering?
Yeah. So in the past, we've focused mostly on the CDR3, but there definitely are interactions, as I showed in that picture, with the other CDRs and the peptide. So I think it would be a good idea to include those, and I think that might improve the, I mean, going back to Alexandra's question, I mean, what we're really interested, I suppose, is identifying the residues that are important, not just in classifying. I mean, we want to know what the features are that are doing the classifying, essentially. And I think the CDR1 and in some cases CDR2 will be of value there. So I think we might as well extend it to those. But again, I think we need to focus on, if we're going to use machine learning, I mean, we want the machine to learn the features. So we're not so interested in classification necessarily as a interpretable machine which actually uses the features of importance rather than features which may be associated with, but not necessarily the key ones that are mediated the interaction. I mean, we want to try and generate biological insight from our machine learning, and that's quite a big challenge. In the past, we often find, as you've just said, that what we get is a highly distributed machine learning tool, which learns a little bit from every position, but doesn't really distinguish the important positions from the non-important positions. So we're trying to introduce features, some sort of more stricter feature selection as we go along to try and get to that question.
Okay, another question from Francesco Camalia. Thanks for the great talk. How would you include information about the V and J usage in the similarity measure?
Well, I mean, we know empirically that that sets of T cells which recognize the same antigen tend to use a subset of Vs. Sometimes they all use one V, but more commonly they use a few Vs. So that information is clearly useful. One of the advantages of the probabilistic network type approaches is that they're super flexible. So you can introduce V and J just as another kind of row of nodes, if you like, another level of nodes. And so you can just, and they will be conditioned on the antigen just as the T cells and just as the amino acids and the triplets are. So from that point of view, I mean, I'm not sure whether your question is mechanistic. How would you incorporate that data? I think that's not very difficult. It's just an additional feature which you can code in many different ways. But if you mean, is that information useful? I think it almost certainly is useful. Yeah.
Yeah, I think it was a mechanistic question, but I think maybe with that we're out of time, and we can find maybe all our speakers in Gather and continue the discussion. And